Okay, so these slides are going to look slightly different than what you already have. And it's not that they're really that much different. I just kind of wanted to make some room for me to draw into things like that. So we're going to talk about movement through the plasma membrane. So this is an animal cell. And the thing about an animal cell is you have to be able to get things in and out of this cell. So we're going to talk about how we successfully do that and the methods that we use to move things. So here's my cell. Inside of the cell, hang on one second. Okay, inside the cell, I have enzymes, I have proteins, I have glycogen, which is our polysaccharide, and I have very high potassium. And this is true for most of the cells in your body. Outside in this extracellular fluid, you've got high sodium, you've got high calcium, and you have high chloride. Okay, this is where I'm getting that from. The thing about your cell is I have to be able to get nutrients out. I have to be able to get wastes. I have to be able to get nutrients in, and I have to be able to get wastes out. Now, why is that such a kind of difficult thing to do? Well, if you think about the volume inside the cell, it really can't change. So if I'm taking something out, I also have to put something in, because if I don't do that and all I do is take out and take out and take out and take out, eventually you're going to end up with something that's shriveled up like a raisin. When it comes to nutrients and getting them in, if I keep filling and filling and filling this cell, eventually it would explode like an overinflated balloon. So there has to be balance. It always goes back to that idea of homeostasis and making sure that for everything in, something goes out. For everything in, something goes out, so that the volume of our cell stays stable. Now, what moves and how? Things that can just basically go through our selectively permeable plasma membrane include things that are lipid soluble and things that are very small and water soluble. If you're small enough to sneak past those tails, it's not that big of a deal, and you just kind of sneak past. But lipid soluble molecules don't really have to worry about the tails not liking them, because they don't like water either. So things like heroin, things like drinking alcohol, things like nicotine, these are all lipid soluble and they bypass that membrane with no problem whatsoever. Now, large lipid soluble molecules and large water soluble molecules actually have to be carried across. They have to have a transport protein. We talked about the uniporter and the symporter and the antiporter. Those actually are transport proteins. They're actually kind of putting them into themselves and then carrying them over. It isn't just kind of a doorway that's wide open. The larger water soluble molecules and even whole cells, these we move via vesicles. Vesicle is a very fancy way of saying bubble. Okay, so we basically take a piece of our cell membrane and enclose something in it to bring it in, or we enclose it in a bubble inside of our cell and then fuse that bubble with our cell membrane when we're moving it out. But in this case, in the third case, we're either talking about large volumes, large molecules, or entire cells, which you will see in just a bit. Okay, so let's talk about the two ways that we transport things. Okay, we've got passive transport mechanisms and we've got active transport mechanisms. Now, simple, passive, no energy is required. Okay, active, energy is required. Those are two really, really big important things. So let's talk about No energy? Energy. Let's talk about how things move without versus with energy. And I know you're probably going, why did she draw a car? Well, I drew a car because it kind of follows the same logic. If I've got a pitcher, of water, right? Pitcher of water. And I have a drop of food dye. And I drop that into the water. That food dye is going to spread out all throughout this pitcher, right? That's actually passive transport. Technically, I don't have to mix the water with the spoon. I don't have to boil it. I don't have to do anything. Literally, it'll just spread out. Okay? Now let me explain. We'll go back to the car. On the hill, this is our hill. And if I could spell hill, that would be better. The car is at a high point on the hill. And if I want to put it at a low point on the hill, do I have to start the engine? No. Technically, all I really have to do is put it in neutral and gravity will make it go from high to low. I don't have to burn any type of gasoline to get this to move. Okay? No energy required. When it comes to passive transport, it's the same concept, but instead of high as in height and low as in height, we're talking about going from a high concentration to a low concentration. Okay? So, if you think about that droplet of food dye, the dye in that droplet is really, really concentrated. That's why when you look at the bottle, it looks like red, right? But then when you put it into pure water, there is no dye in this pure water. So that high concentration of dye here spreads into the low concentration of dye in the water because it's pure water, right? So there's no dye there to begin with. That would be like saying ground level versus the top of the hill. So with passive transport, you're going from a high concentration to a low concentration, and the movement does not require any energy. So that's passive. Now let's talk active. Okay? So in active transport, we're actually doing the exact opposite. Okay? So in active transport, we're starting off at a low concentration, at the low part of the hill, and we want to put the car up here at the high part of the hill. Now, if I want to take the car from here to there, do I have to start the engine? Because if I put it in neutral, is it just going to whoo and go up to the top of the hill? No. So, in this case, I have to have energy because I'm moving from a low point to a high point. So, when you talk about active transport, you're talking about moving from a low concentration to a high concentration. So remember that beaker I was just talking about, that beaker with the food dye in it, right? So the dye is all throughout here. What if I wanted to go from this and put the dye back in the droplet? So low concentration to high concentration, right? How do we do that? Well, I mean, I guess I could take a distillation system and use that. But in all honesty, distillation systems take energy. Same, I can do something even simpler than that. I can put it in a pot on top of a burner on a stove and let the water boil off. The thing is, even doing that, I'm still putting energy in because I'm using the fire or the electrical burner or whatever. So in order to go from a low concentration up to a high concentration, you actually have to put energy behind it. It doesn't just automatically happen. Okay? And that's active transport. Okay? So we've got several passive transport mechanisms we're going to talk about: diffusion, osmosis, and facilitated diffusion. And we've got several active transport mechanisms we're going to talk about: primary active transport, secondary active transport, and vesicular transport. That includes endocytosis and exocytosis. So let's start by talking about passive transport. Now remember, passive transport means I'm going from high to low, and there's no energy required. Required. That is not spelled right. Required. There we go. Okay, so let's start with passive transport. The first mechanism that uses passive transport that we discuss a lot in A&P is diffusion. Diffusion is the movement of solutes from an area of high concentration to an area of low concentration. Solutes would be things like sodium or sugar or chloride for that matter, or chlorine, I should say, for that matter. Anything that would dissolve in a solution. So we've got this movement from a high concentration to a low concentration. Now, the thing with diffusion is we're always talking about solutes. Okay, that's a big thing. We're always talking about solutes. So the concentration gradient. So what's a gradient? If you're talking about an actual hill, this has kind of a small gradient. This has a large gradient. It's the difference between the top and the bottom of that hill. Okay, so the distance between the top and the bottom here is like that. Difference between the top and the bottom here is quite a bit larger, right? So the concentration gradient is the difference between the low point and the high point. If you have high, not height, high point, if you have a huge distance between them, like this, then you have a really high concentration gradient. If you don't, then it's not that high. I mean, it is a concentration gradient, don't get me wrong, but it's not a high concentration gradient. Okay? So, what can change how fast something diffuses? What can change how fast something goes from being up here on the hill to being down here on the hill? Well, one of them is the magnitude of the concentration gradient. How big is that difference? If I've got a car on top of a hill like this versus a car on a hill like this, which one is going to go faster from top to bottom? This one. Why? Because that difference is a lot bigger. From here to here, that car is going to, right? As opposed to from here to here, it'll still go, but it's not going to be near as fast. So how big is the difference? Another thing, and we did kind of already talk about this once before, we talked about temperature, the temperature of a solution. So anytime that you increase the temperature of a solution, you're causing the molecules in that solution to move around faster. Molecules in everything vibrate. The molecules in you right now are vibrating. But when you heat them, they vibrate even faster. And ultimately what that means is when you heat something and those molecules are kind of bouncing around faster and faster, it helps to spread whatever it is you're trying to diffuse even faster. The best example I can think of, you've got a cold glass of tea and you've got a hot glass of tea, and you put 3 teaspoons into each one. Which one does that sugar dissolve in faster? The hot tea, right? And honestly, one of the main reasons is because of this. Because we put the same amount of sugar in both, 3 teaspoons in both, but having that temperature difference actually helps to spread and diffuse that sugar through the solution. That's why when we make sweet tea, we tend to do it when it's still hot. Now, the size of the diffusion molecule. Okay, so if I have a swimming pool that is the size of the building that you're in right now, and I put, let's say, 15 pounds of sugar into that swimming pool. Sugar's going to dissolve, right? I mean, it'll go throughout the whole entire swimming pool. However, if I take 15 pounds of bricks, let's say that each brick weighs a pound, and I dump them into a swimming pool, are they going to spread out? No. Why? They're too big, they're too heavy. They don't just go wherever. And to be honest, it's the same. Yes, we may be talking about something that has more of an atomic mass size to it, but ultimately the size still dictates whether or not this thing will move. And finally, the viscosity of the solvent. Viscosity refers to the thickness. Okay? So water is not very viscous. If I put water in a pitcher and I pour it, it just goes. There's no kind of stopping it. However, if I were to take pancake syrup and do the exact same thing, put it in a pitcher and try and pour it, it's not like the pancake syrup pours as quickly as water. It's kind of slower and it's kind of thicker. That's viscosity. So ultimately, it's kind of the same thing. I may be talking about the temperature of what we're dissolving in, like the water, the hot versus the cold. I may be talking about the size of the thing that I'm dissolving, the sugar versus the bricks. But ultimately, even when it comes to that thing that I'm trying to dissolve inside of, if it's thick and viscous, it's not going to let that stuff spread like it's supposed to. In fact, it's going to impede it from doing that. So, diffusion is the movement of solutes, salt, sugar, whatever, but it's the movement of the solutes. Osmosis is still a type of diffusion, but now we're talking about water moving. So we're not talking about salt or sugar moving. Now we're talking about water going from a high concentration to a low concentration through a selectively permeable membrane. Selective, I decide who gets to go through. Permeable, if you fit, you may go, but if you don't, you're not going anywhere. So, a selectively permeable membrane normally will let water go through, but not the solutes. So remember how over here we said diffusion is the movement of solutes? Yeah, now they can't move. So our cell membrane, our plasma membrane in our cell is very much a selectively permeable membrane. So water and its movement into or out of our cell is actually fairly important because it does kind of decide where things move. Okay? So if you look here, all right, this little Erlenmeyer flask looking thing has an open bottom, but I've put this selectively permeable membrane around it. If you look inside of here, it's a 3% salt solution. And you can see I've got the green and the pink, the sodium and the chloride, and there is water in there. You can see the little blue dots that make water, the same blue dots that are out here. But look at how many blue dots are here versus in the beaker. When it comes to osmosis, water wants the same number of water molecules out here as it has in here per salt molecule. Well, what's it going to do? It's going to start going through this selectively permeable membrane, basically to try and find that balance. Now, can it find that balance? No, it can't. This can come up here like this, but that salt never finds its way out because it doesn't fit through that selectively permeable membrane. So if you look down here at the bottom, you'll notice that when I put this down into the water level starts to rise here. And you can see that water level gets almost to the end here, right? That movement of water from a high water concentration to here where it's a low water concentration does take place, but eventually it stops. It's not like this overflows like a fountain. Now, why this? Eventually, the weight of that water creates something called osmotic pressure. It gets too heavy. It would be like me saying, yeah, I can lift a 10-pound weight. Yeah, I can lift a 20-pound weight. Yeah, I can lift a 40-pound weight. Yeah, I can lift, but eventually I'm going to get to a point where I'm going to be like, yeah, I can't lift that anymore. Okay? So that is called osmotic pressure. Remember, osmo is water, so water pressure. It's the force required to prevent water movement across a selectively permeable barrier via the process of osmosis. So what we're seeing here is literally osmotic pressure is stopping water from moving anymore. Okay? Now, in chemistry, we tend to use the terms isoosmotic and hyperosmotic and hypoosmotic to kind of describe solutions that you can put these selectively permeable membranes into. But in the human body, we don't really use those. We use a different set of terms. We use tonicity. So, tonicity is literally referring to your cell's shape remaining constant because that internal pressure inside of your cell, hang on, the internal pressure inside of your cell is stable. Okay, so tonicity means that you've got this constant pressure, internal pressure inside your cell pushing out. Now, for the sake of argument, we're gonna go with something fairly simple, and these aren't real numbers, but let's say that inside of my cells I have 50% water and 50% salt. So 50% water, 50% salt. And again, these are my cells. 50% water, 50% salt. If I put a cell into a solution that is also 50% water and 50% salt, and we're talking about water movement, don't forget that. We're talking about osmosis. So if we're talking about osmosis, that 50% water here and the 50% water in the beaker are equal, right? They're the same. So this solution that my cell is sitting in is exactly what is inside of my cell. So we call this an isotonic solution. It's the same. Remember, iso means the same. So in an isotonic solution, because again, water wants everything equal. If I, hang on a second, get a pen. Okay, if I lose a water molecule, I'm going to gain a water molecule. If I lose a water molecule, I'm going to gain a water molecule. It's all about equilibrium. You want it to be isotonic, okay? So it's at that point that no matter what I do, because I want it equal on both sides, and I've got it equal at both sides, if anything moves, something else is going to move the opposite direction. So let's talk about a hypotonic solution, okay? In a hypotonic solution, you will have something like 70% water and 30% salt. Okay, again, made up numbers, but it gets the point across. So, if you look at the water concentrations, it's 50% inside my cell, and it's 70% in the beaker. Which is the bigger number, 50 or 70? 70, right? So, it's going to go from in my beaker to in my cell. So it's going to start flooding into my cell. Now, mind you, they're not balanced, right? So 70 and 50, I want to get it 60-60 so that it's the same on both sides of this membrane. So I keep shoving water into this cell. Now, if you're really lucky, it'll be close enough that it'll balance and everything will be okay. When you're not, though, the water rushes into your cell until it overinflates like a balloon, and eventually it can actually split. It can actually explode. When that happens, we say that the cell has lysed. Okay? Which you can see in number 3 up here. Lysis is when your cell basically ruptures because you overfilled it. If you've ever been in the hospital and had an IV put into your arm, the reason that they don't use pure water in that IV and instead use saline is for this reason. If they were to put pure water in your IV, all of your cells would rupture. So again, not a good idea. Now, let's go the opposite direction of that, okay? Let's go to a hypertonic solution. So, in a hypertonic solution, you would have something like 30% water. Let me write that over here. 30% water and 70% salt in the beaker. Looking at the numbers, in my cell I've got 50% water. In my beaker I have 30%. Which number is bigger, 50 or 30? 50. So the water is going to move from my cells into the beaker, because this is the beaker concentration. So literally, the water starts leaving my cell, and my cell starts to shrivel up like a raisin. That balance that we count on for tonicity is not there. This can also kill your cells. Okay? It can also destroy your cells. So when this happens to your cells, we call it crenation, which you can see up here in number 2. So that osmosis, the power of osmosis and the ability of our cells to maintain their internal pressure is really important. And if you don't have the proper balance, you can really do some damage to a body. So the last type of passive transport we're going to talk about is facilitated diffusion. Now notice, diffusion's still part of it, so I'm still moving some type of solute. But normally with facilitated diffusion, facilitate means to help. With facilitated diffusion, you're talking about moving something a bit bigger. So you're not talking about, let's say, a sodium ion anymore, one atom. Now you're talking about moving an amino acid, or you're talking about moving glucose. You're talking about moving molecules not just one ion or one atom. Now, I can't move those directly via diffusion because they're too bulky. What I can do is I can create a transport protein. We just talked about uniporters and symporters and antiporters, right? I can create a transport protein that basically helps me to get to that point of equilibrium. Remember that with diffusion, this is still diffusion, no energy is required because I'm going from high concentration to low concentration. Okay, so using mediated transport, again, this is kind of helped out transport. I have a protein that helps me to kind of get equal on both sides. If you look at this example, technically I have 4 glucose here, right? But then if you look on this side, I've got 1 and a second coming in the same direction, and then I'd have 1 and 2 on the opposite side. So I'm not moving everything, I'm just trying to get it equal. You don't drive a car off a hill and it ends up under the ground. You drive a car off the hill, and once you're level, it levels off, right? Okay, so those are the mechanisms of passive transport.